So I'm a transplant hepatologist, which means that I treat patients with very advanced forms of liver disease who have required a liver transplantation. And from a scientific standpoint, my passion is to try to understand how the immune responses within the liver are regulated, and in particular how we can inhibit rejection without resorting to administering lifelong immunosuppression. So transplantation is without any doubt one of the greatest success stories in medicine over the past 60 years. The shortened outcomes after a liver or kidney transplantation are exceptionally good, and this is due to advances in surgical techniques, but very particular to the discovery of very powerful and non-specific immunosuppressive drugs. However, what people are not so aware of is that in the long run, the outcomes after a liver or kidney transplantation are actually not that good, and they remain quite suboptimal. For instance, in liver transplantation, the long-term survival is significantly lower than that of the general population, but it's also lower than the survival of non-transplant patients who have severe diseases, such as, for instance, breast cancer. And there are several reasons why this is the case, but without any doubt, one of the key contributors are the side effects of these non-specific immunosuppressive medications that transplant patients need to take lifelong in order to protect their grafts from the harmful effects of rejection. So in this regard, transplantation is not very different from what happens in patients with very severe forms of autoimmunity, who also need to take lifelong immunosuppression and who also have poor long-term outcomes. So it's clearly one of our greatest challenges here is to identify new strategies to inhibit rejection in a very selective and permanent manner, so without affecting immune responses that are not directed against a transplanted organ, and without requiring the lifelong administration of very toxic medications. Clearly, one of the probably most extraordinary advances I have witnessed over the past 2 decades is the development of tools that allow us to very selectively boost the immune system to kill cancer cells. This has resulted in what look like miraculous complete remissions in terminal cancer patients, and is the reason why immunotherapy is has taken nowadays the central stage in oncology. Now, from a scientific point of view, what is absolutely fascinating is that despite the fact that there is a lot of redundancy in the immune system, it is possible to very selectively target key molecules or immune cell components of the immune system, resulting in huge clinical benefits. And this has incredible implications for diseases other than cancer, such as autoimmunity or transplantation, because many of these targets are actually shared by many different immunological processes. So regulatory T cells, or T-regs as we call them, are a small subset of T lymphocytes that have intrinsic immunosuppressive properties, and that they are essential to prevent autoimmunity and to establish what we call immunological tolerance. Now, if humans lack regulatory T cells, they die due to severe autoimmunity, and in animal models, if we eliminate this cell subset, then the immunosuppressive properties of many different types of therapies are completely abolished. So one of the reasons why regulatory cells are so attractive from an immunotherapeutic point of view is the fact that they can be grown in the laboratory in large numbers without losing their suppressive function or their phenotype. And this opens the door to treating autoimmunity or to preventing rejection after transplantation by administering large numbers of ex vivo expanded regulatory cells. Now, there is a further reason why T-regs are very appealing for immunotherapeutic purposes, which is the fact that they can be genetically engineered so that they acquire more powerful suppressive function, or they develop a specificity against predefined antigens. And this is precisely what we have established at Quell Therapeutics, with the vision of treating inflammatory disorders, autoimmunity, and also preventing rejection after transplantation. Now, we believe that this strategy will deliver very significant clinical benefits to a variety of immune-mediated diseases in which the current standard of care is clearly suboptimal. And furthermore, what is very exceptional about regulatory T-cell therapy is that because Tregs are so important, so central to the pathogenesis of many immunological diseases, there is the promise for the first time to be able to completely modify the way the diseases are being managed by providing a permanent cure. Syncona have a very impressive track record of partnering with academic investigators to create successful biotech companies, and in particular, they have acquired quite a substantial expertise in what is the industrial development and the commercialization of cell therapies. As the founding team, on the other hand, we provide expertise in Treg biology, in Treg engineering, and also in the performance of clinical trials using ex vivo expanded regulatory T cells, and not only in transplantation, but also in autoimmunity. With Syncona has been both intellectually challenging and very gratifying. It is a challenge because we need to bring industry standards into our routine work, which means that often we need to modify or adapt our experimental practices. It is also challenging because it forces us to keep in mind at all times that we need to develop commercially viable products if we are to really transform clinical practice. And this is something that we academic investigators are not used to do in our routine activities. Now, it is very gratifying, however, because we are working with individuals who share our vision and who are absolutely committed to develop it, and who have the intellect and, more important, the financial resources to make it happen. So our current project is very much centered around the idea of generating a cell therapy product that will be able to induce immunological tolerance in the setting of liver transplantation. We think that this is a fantastic opportunity to provide a very transformative clinical effect, but also a very unique clinical setting to actually test the biological efficacy of these genetically engineered regulatory T cells.